Hello, myself Kainde HG, Department of Zoology, Daivadi College, Daivadi. Today we discuss about the replication. In DNA replication, we all know central dogma of biology. Central dogma of biology: replication, transcription, and translation. In Tamin and Baltimore, they discuss or propose the reverse transcriptase enzyme. They show the replication is transferred to transcription. Again, mRNA can be transferred into DNA. In replication, there is a duplication of genetic material. Again, in there is a transfer one strand converted into second strand. We all know DNA is a biological process. Okay, in there single strand converted into double strand. Okay, there is a transcription after next process of the replication. Next, this is all the process of the DNA replication. DNA is we all know that DNA is a double helix structure. DNA structure proposed by the Watson and Crick. So Watson and Crick proposed the DNA structure. They firstly they mistake on the because DNA is the leading strand. Okay, there is a continuously replication are there, but it's not true. There is a discontinuous type of replication occur. There is the single strand against single strand. Here the replication machinery are available. In replication, next slide we show the replicated machinery. DNA is we all know DNA is a complementary strand. Means there are the complementary strand, means their parent and daughters are show similarity in their strands. Okay. During replication, these strands are separated. Okay. During replication, these strands are separated, and again new strand are synthesis. Origin of DNA molecule then act as a template. Okay. Any strand, in both of them, any strand act as a template strand. Next. General features of replication. First one, template dependent process. Any synthesis of DNA, there is a requirement of template. Okay. Without template, there is a no replication. Okay. That reason, DNA's replication is the template dependent process. Okay. Second is always occur in 5' to 3' direction, because there in replication primer are provided. Primer are the 3' to 5' direction. Okay, there is just 10 nucleotide to 16 nucleotide primer are provided. Because of, again, one of the reason behind that, which one? They are the proofreading activity are there. Okay, if DNA direction, replication direction occur in case of 3' to 5' direction, then there is no perform of the proofreading activity. But what main question is that what is the proofreading activity? Proofreading activity is the backspace of the nucleotide sequence. Okay, there is a clearance of nucleotide sequence. Okay, that reason mutation. If proofreading activity actually work, then mutation less can occur. Okay, that reason always occur replication occur 5' to 3' direction. Okay, again behind reason there is a polymerase activity. Okay, polymerase enzyme work. Polymerase enzyme is a work in one direction. That reason replication 5' to 3' direction. Again third one, DNA is a semi-conservative in nature. Okay, what is meant by semi-conservative in nature? There is a two strands form after the replication. There is a one is a parent type and second is a daughter type. Yes. Then again type of nature of DNA is a discontinuous. Semi-conservative, conservative. In conservative type of replication, there is a as it is type of parent type of DNA strand are found. Again third one Third one is the disruptive type of nature. Okay, what is in the disruptive type of nature? Disruptive type of nature are small strands are formed one after one, like parent strand, then daughter strand, then parent strand. Okay, this type of synthesis they call disruptive type of nature. But in case of eukaryotes, prokaryotes, there is a semi-conservative in nature. Again, bidirectional. In case of virus, plasmids, there is a DNA replication is the unidirectional. But in case of eukaryotes, the DNA replication is a bidirectional. There is a different type of machineries are work. Again, DNA replication property. DNA replication is the semi-discontinuous. What is the semi-discontinuous? Semi-discontinuous, there is a two strands of DNA of synthesis. First one is a leading strand, second is a lagging strand. But in case of lagging strand, there is a large primer are required. Lagging strand, DNA replication is a slowly. That reason, DNA replication is a discontinuous. Okay, again, in replication space, DNA replication begins at specific location, origin of replication in the genome. What means is that DNA replication requires override. Okay, or in case of eukaryote, there is a ORC. Okay, in case of prokaryote, there is a single override. Or in case of virus, in case of plasmid, there is a single override. But in case of eukaryotes, there is a thousands of override. There is a start of replication. Again, second, origin of synthesis of new strand accompanied by the enzyme known as helicase. We all know that there is a replication machineries. Different enzymes are work there. They help to replication proceed out. Again, these are the protein machinery. They work in the both prokaryotes and eukaryotes. First one is the DNA protein. In case of eukaryotes, they are homologs. These are the proteins work in the prokaryotes, and these are the proteins work in eukaryotes. We can say these are the homologs each other. First one, DNA protein works in prokaryotes when DNA are totally methylated form. Okay, there is a 13-month and 9-month sequence. In 13-month sequence, lot of double bond there. There is a melting of DNA. Okay, when melting of DNA occur, then DNA protein are work. Okay, then DNA protein we can call them helicase loader. Okay, what work a helicase loader done? They are load the helicase on the DNA double strand. Okay, in case of eukaryote, DNA we can call MCM10 or licensing factor. Okay, there is again a nomenclature are CDC6 and CDT1. We call them licensing factor. These are work as it is. Okay, then DNA act as a helicase in prokaryotes. Again, as it is homolog in eukaryote, MCM227. Okay, but in case of prokaryote, DNA be load on a single strand DNA. Okay, but in case of eukaryotes, MCM227 load on a double strand DNA. But when it loaded, they are in active form. Okay, when in case of DNA, it's a totally active form. They work in an active form. Okay, then again, next protein is a SSBP. Okay, SSBP work as a denaturation process to renaturation process stop. Okay, in case of eukaryote, as it is work as a RPA. In case of when helicase separate the two strand, after some time it renaturation process again occur. That reason SSBP hold the two strand to opposite each other. Their role performed by SSBP, and as it is in eukaryotes RPA. Again DNAG act as a primase. Okay, and in case of eukaryotes, alpha DNA polymerase act as a primase. But what is the primase? When replication are start, there is a need of small nucleotides strand. We call them primer. Okay. Topoisomerase, and as it is rolling eukaryotes, there is a two type of topoisomerase. Firstly, one is topoisomerase first and topoisomerase second. Topoisomerase first work on a single strand DNA, or topoisomerase second work on a double strand DNA. It can help the replication to negatively supercoiling form. Okay, we all know DNA is the negatively supercoiled form. Then it can replicate. Okay, then again there ligases. Ligases can help in the sealing process. Okay, when DNA replication are completed, then ligases are sealing process help. Topoisomerase also help in decatenation process, which help to replicate replication of DNA. Replication are divided into three stages. Which one? First one is the initiation, second one is the elongation, and third one is the termination. Replication process three enzymatical catalyze and coordinate steps: initiation, elongation, termination. During the initiation, protein bind to origin of replication. Up to helicase perform, there is a initiation step. Up to helicase perform, there is a initiation stage. Next one is the elongation. Elongation, primer sequence are extended in elongation process. After elongation, nucleotide can add and synthesis are done. After termination, elongation, there is a termination. Termination end replication problem are created in the termination. Which one? Linear form of DNA. Okay. In case of eukaryotes, there is a termination problem. In case of prokaryotes, there is a ter site, and ter site there is a tus and ter protein are one. Okay. Thank you.